Hi, in this class we'll study the morphology of bacteria. So we are going to study the shape and size of the bacteria, structure of a typical bacterial cell, the spore structure and the process of sporulation. The bacteria are measured in terms of micrometers. So we know a micrometer is a 1,000th of a millimeter. A nanometer is a 1,000th of a micrometer. Angstrom is 1/10th of a nanometer, and the size of a bacteria is measured with the help of the micrometry. So micrometry is a microscopic based measurement of the size of the objects, so especially the bacteria. So bacteria varies in their shape, and also knowing the size and shape of the bacteria will help us to presumptively identify which bacteria it can be. So the shape of the bacteria, so we have a cocci, which is derived from the word coccus, which means a berry. So you might have seen a berry. So it's a spherical structure. So in this, the cocci, the length is equal to the breadth. So it is a spherical structure. So we know, even though it looks two-dimensional in this picture, so the actual structure in the world, it will be a three-dimensional structure. Bacilli, it is derived from the word baculus, which is nothing but a rod. You can see this is a rod shaped structure. Okay, so here the length is more than the breadth, so that is why this is called as a rod shaped bacilli. Okay, so vibrio means a curved rod. Actually, it is the word vibrio is derived from the vibratory movement it does, but it's a curved rod. The spirilla is a rigid spiral bacteria, whereas spirochetes are the flexuous spiral forms. Okay, the spirochetes means spira means coil, chite means hair. It's a hair-like coiled bacterium which is called as spirochetes. The another shape of a bacteria is actinomycetes. This is a branching filamentous bacteria. The terminology is derived from two words, that is, actis means array of cells. You can see, if you consider this is the center of this structure, and you can see all these filaments are arising from the center and they are radiating. So it looks like a ray of sun. And myx means fungus, because filaments which is a similar shape of that of the fungus, so they were called as a sun ray like appearance and also a fungal morphology. They were called as the actinomycetes. So the last one is pleomorphic bacteria, which can be in the form of like Mycoplasma, which does not have a cell wall, because the cell wall is the one which is going to give a structure to the bacterium, and we're going to study about the cell wall in the subsequent slides. So bacteria like Mycoplasma, Haemophilus, and some of the anaerobic bacteria, they occur in different forms, and they are known as the pleomorphic forms. Now, after studying the size and shape, the arrangement of bacteria is very important. So as I already told, the size, shape and arrangement, all these three helps us for the presumptive identification of the bacteria. Okay, so if a cocci are arranged in pairs, it is called as diplococci. If they are in 4, they are called as tetrads. If they are a cluster of 8, they are called as sarcina. And if they are in an irregular structures and like a bunch of grapes, it's called as the staphylococcus. So if the same cocci, they are arranged in chains, they are called as streptococcus. Similarly, if bacilli are arranged in chains, they are called as streptobacillus. So if the bacilli are arranged either in parallel or in like a X, V or L forms, they are called as the cuneiform arrangement or the Chinese letter pattern of arrangement. So this is an example to show the bacilli and cocci. The stain used in this slide for the slide is the gram stain. We can see gram negative bacilli and gram positive cocci. Now we will come to the structure of a typical bacterial cell. A bacterial cell, it is having the bacterial cell envelope. Inside is the protoplasm, and outside the bacterial envelope, attached to it are some of the surface appendages. The bacterial cell envelope includes the cell wall and capsule, periplasmic space, and the cell membrane. Protoplasm contains mesosomes, rhizosomes, inclusion granules, vacuoles, or nucleus with a single circular DNA, but it won't be having a nuclear membrane as with that of the eukaryotic bacteria. The surface appendages in some of the bacteria, it can be a flagella or pili. So this is just a schematic diagram of the structure of the bacteria. Let's take up the cell wall. Cell wall is a rigid structure which covers the bacteria. It's a chemically complex structure. Because of its rigidity and complexity, it gives a proper shape to the bacteria, and it also protects the bacteria from the external environmental insults, such as the change in pressure or sometimes the movement of ions and many other things. Chemically, the cell wall, that is the outermost layer, it is composed of N-acetylglucosamine and N-acetylmuramic acid, which are joined together by short peptide chains. So N-acetylmuramic acid carries a pentapeptide chain. You can see this is an N-acetylmuramic acid, N-acetylglucosamine. So N-acetylmuramic acid is having a pentapeptide chain. So the pentapeptide side chains are of a two different N-acetylmuramic acid in two different chains. They will be cross-linked by pentaglycine bridges. So the pentaglycine bridges, they cross-link the two N-acetylmuramic acid of the peptidoglycan chain. So based on the differences between the cell wall of the bacteria, we can classify the bacteria into gram positive and gram negative bacteria. So I am going to brief some of the salient features of the gram positive cell wall and also the gram-negative cell wall. Then later on we will discuss the ultrastructures of the each. So gram-positive cell wall is a thick structure of 15 to 80 nanometers, and this is a homogeneous structure. So what I mean the homogeneous means it is composed of similar kind of chemical molecules. It contains large amount of the peptidoglycans which are present in several layers, and also the peptidoglycan layer constitutes up to 80% of the dry weight of the cell wall. It also consists of the teichoic acid and teichuronic acids, which may account up to 50% of the dry weight of the cell wall, or 10% of the dry weight of the total cell. The peptidoglycans are connected to each other by the covalent bonds. So this is the gram-positive cell wall. This is the cell membrane, which is nothing but the phospholipid layer, and after that this is a layer of the peptidoglycan chains arranged in several layers. Okay, so we can see the cross-linking teichoic acid, and those teichoic acid which starts from the cell membrane and reaches all through the peptidoglycan layers, they are called as the lipoteichoic acids. Some of the surface proteins will be there, but the protein content as such will be less in case of the gram-positive bacteria. The teichoic acid are the polymers of the polyribitol phosphate or polyglycerol phosphate containing the ribitol and glycerol. Okay, so they have the sugars or amino acid side chains, and there are two types of the teichoic acid. The wall teichoic acid, which I told, which will be there, attaches the different layers of the peptidoglycan layers, that specifically the N-acetylmuramic acids, and lipoteichoic acid, it attaches from the cell membrane to the peptidoglycan layer. So both this teichoic acid gives some kind of holding the cell wall to the cell membrane, as well as holding the different peptidoglycan layers together. The next structure is teichuronic acid. This is nothing but the repeating units of the sugar acid, that is N-acetyl manuronic acid or N-acetyl glucuronic acid. So they are synthesized in place of the teichoic acids when the phosphate supply to the cell is very much limited. This is the diagrammatic picture of the gram-positive cell wall. Gram-negative cell wall, the amount of peptidoglycan layer is very small, say around 2 to 8 nanometers when compared to the up to 80 nanometers in case of the gram-positive cell wall. So, what I mean to say is the thickness with respect to peptidoglycan is very less. Otherwise, the cell wall of the gram-negative bacteria will be large with respect to the lipopolysaccharides which are there in the cell wall. So now we'll see outside the peptidoglycan layer there is a layer of lipoprotein, there is an outer membrane, and also the lipopolysaccharides. So lipoproteins are covalently joined the peptidoglycan layer with the outer membrane. We will see the structure in the next slide. So these lipoproteins, they stabilize the outer membrane of the gram-negative cell wall. The outer membrane is a bilayer structure. The inner part, which is similar to that of the cell membrane, and the outer part is containing of lipopolysaccharides. The outer membrane is will be having the porins, which is nothing but special channels which permit the low molecular weight hydrophilic compounds such as the sugars and amino acids and ions from the external environment to the inside the bacteria. And also they exclude the hydrophobic molecules, and these porins, they serve to protect the cell. The outer membrane proteins, outer membrane proteins C, D, F, PHOE, LAMB are the major proteins which are responsible for the transmembrane diffusion of the nutrients. Sometimes the outer membrane proteins like TSX, which acts as a T6 bacteriophage receptor. The outer membrane protein A anchors the outer membrane to the peptidoglycan layer and also helps in the conjugation process. The outer membrane proteins are involved in the transportation of the vitamin B12 and iron. The lipopolysaccharides are the complex molecules which consist of the three main components. One is the lipid A. Okay, lipid A is nothing but the phosphorylated glycosamine disaccharide along with the chains of the fatty acids attached to the glycosamine. Okay, it consists of the hydroxy myristic acids, which is an unique fatty acid, and this lipid A is associated with the endotoxic activity. The core oligosaccharides include two characteristic sugars, that is ketodeoxyoctanoic acid and an heptose, which join the lipid A to O polysaccharides. Usually the core oligosaccharides are genus specific structures. The lipo-oligosaccharides, they are short multi-branched glycans present in the bacteria like Neisseria meningitis, Haemophilus influenza, that colonize the mucosal surfaces. They exhibit an extensive antigenic and structural variation which can act as a very important virulence factor for the bacteria to survive in the host environment. It has an terminal N-acetyl lactosamine residue which has a molecular mimicry of the host antigen. The O polysaccharide extends from the core oligosaccharides, and there are species specific ones, and variation helps the bacteria to survive in the environment. So if the bacteria does not have the O polysaccharide, it forms what is called as the rough forms of the colonies, and if it has the O polysaccharide, it forms the smooth form of the lipopolysaccharides. Now to differentiate, so we can have this gram positive and gram negative cell wall differences. Thickness of peptidoglycan is around 15 to 80 nanometers in case of gram positive, 2 nanometers in case of gram negative. Lipid content is just 2 to 5% in case of gram positive, 15 to 20% in case of gram negative. Teichoic acids are present in case of gram positive, absent in gram negative. There are variety of the amino acids, and the aromatic amino acids are few are absent in case of gram positive. There are several in case of gram negative, and the action of the cell wall as endotoxin is often seen in case of the gram negative, and treatment with the lysozyme, with the gram-positive cell wall or gram-positive bacteria, they become protoplasts, and gram-negatives, they become spiroplasts. Let's see, after studying the gram-positive and gram-negative cell walls, let's see a brief differences in the what is the composition of the acid-fast bacilli. Actually, they are also composed of the peptidoglycan layers, but there is an association of asymmetric lipid, what is called as the lipoarabinoglycans. So the lipid bilayer contains of the mycolic acids linked to the arabinoglycans, which is called as the lipoarabinoglycans, and the lipid lipoarabinoglycans, they are hydrophobic and they render the acid fast bacteria to be resistant for most of the harsh chemicals, and also this is the structure which gives the acid fastness to the bacteria. So we can see the acid fast cell wall. After the cell membrane, we have a layer of peptidoglycans, okay, so maybe several layers, and then arabinogalactans and the mycolic acids. So lipoarabinomanans are the ones which acts like a teichoic acid in case of gram-positive bacteria, and they connect entire structure to the cell membrane, and they help in the attachment or the retaining of the acid fast cell wall over the bacteria. So to make understand, so I'm going to put all the three types of the cell walls together. So you can, if you want, so you can comment on the comments saying that if you want an extra information about each type of the cell walls, I'm going to take a separate class for that. So after that, the gram-negative bacteria, after the cell wall, they have a space what is called as the periplasmic space. This space will be between the cell wall and cell membrane. Usually this space in case of gram-negative bacteria is filled with a loose layer of peptidoglycan, and also there will be the hydrolytic enzymes and also some beta lactamases and some of the nutrients which are acquired from the external environment. Let's take up few of the atypical forms. So cell wall deficient forms, usually the cell wall removal with the help of either lysosome or blockade of the protein peptidoglycan synthesis, like in case of treatment with the beta lactam antibiotic, that lead to the cell wall deficient forms. Usually in case of chronic infections where we suspect or where we treat the patient with the cephalosporins, there the cell wall deficient forms will arise, and they cause the chronic infection. But once the inhibition of inhibitors of the cell wall are taken out, so that leads to the resynthesis of the cell wall, and the bacteria regains their original shapes. Protoplast is a bacteria which is produced from the gram-positive cells on treatment with the lysosome, and spiroplast is a bacteria which is derived from the gram-negative bacteria when they are treated with the EDTA. So osmotically, both protoplast and spiroplast, they are fragile. Mycoplasma is another structure which is atypical forms. So usually they are naturally occurring bacteria without the cell wall, and they are pleomorphic. L forms, these are the bacteria which are named after they were first identified from the Lister's Institute. They don't have any regular size or shape, and usually they are produced from the Streptobacillus moniliformis bacteria on exposure to the penicillin, and they also produce the chronic infections. So some of the pleomorphic bacteria, so they have considerable variation in the size and shape. So like example is Yersinia pestis. Involution forms are usually seen in the aging cultures, and these involution forms, they will be seen as a swollen or aberrant forms. Usually the high concentration can produce the involution forms. The cell wall can be demonstrated with the help of the differential staining procedures like gram stain, electron microscopy, plasmolysis, microdissection, mechanical rupture of the cell, serological, and also by serological test. Cell membrane is a semi-permeable membrane which acts as an osmotic barrier. Basically, it is composed of the phospholipids and also the proteins. So cell membrane is very important for the production of the energy, because as we know, the ATP is synthesized in the mitochondrion, whereas here the lack of mitochondria, the electron transport chain and the oxidative phosphorylation takes place at the level of the cell membrane in the form of a mesosome, which is in a modified form of the cell membrane, where the ATP synthesis will occur. So it differs from the phospholipid bilayer of the eukaryotic membrane in lacking the sterols, and that is why if we target the phospholipids of the bacterial cell membrane, we are not going to harm the human or the human cell membrane. Cytoplasm is a colloidal suspension of a variety of organic and inorganic solutes. 70% of it is made up of water, and so it is having all the biosynthetic components which are required for the cell division and also the metabolism of the cells. Nucleus, here this is a prokaryotic nucleus. It will be in the form of a single circular double-stranded DNA. No nuclear membrane, no nucleolus, and it is usually haploid, and it multiplies by binary fission. So in the cytoplasm we have extra chromosomal DNA materials in the form of the plasmids. So that finishes the part 1 of the morphology of the bacteria. We will be studying the morphology, the structure of the spore, the sporulation structure in the next class. So thanks for watching the class. Okay, please subscribe my channel and press the bell button to get the updates of the new videos. Thank you.